Hi, my name is Xiao Chen. I'm a bioinformatics scientist at PacBio. In this presentation, I'll be talking about using a PacBio tool called Paraphase to genotype highly similar paralogous genes. The human genome contains a large number of long DNA sequences that are present in multiple copies with high sequence similarity. Many medically relevant genes fall into these so-called segmental duplications, where read alignment and variant calling are difficult due to the presence of gene paralogs or pseudogenes that are highly similar in sequence. To further complicate the problem, segmental duplications are dynamic regions where unequal crossing over can happen, resulting in copy number changes, inversions, as well as gene conversions, which is a process that exchanges the sequences between a gene and its paralog. It's important to detect all of these events in segmental duplications. To resolve the variant calling problem in genes with highly similar paralogs, we developed a tool called Paraphase. The idea behind it is that genes and their paralogs can be analyzed as if they are different copies of the same gene. In this diagram, the green box is our gene of interest, and it has a highly similar paralog in magenta. Read alignment, denoted by those short horizontal lines, are ambiguous and often wrong in these genes. Paraphase extracts reads from all relevant regions and realigns them to the gene of interest. It identifies variant sites and uses those sites to phase reads into haplotypes, which are complete copies of genes denoted by the long horizontal lines. Each haplotype or gene copy is analyzed to identify variants. A copy number change can be called from the number of haplotypes identified for a gene. For example, 3 magenta copies here indicating a copy number gain. We can annotate each haplotype as functional or non-functional and calculate the copy number of the functional gene in a sample and infer the disease or carrier status of the sample. An example of a medically important gene resolved by Paraphase is SMN1. This is the gene where variants can cause spinal muscular atrophy. SMN1 has a paralog called SMN2, which has minimal function. The two genes are near identical in sequence, with just one functionally different base throughout the 30 kb long gene body. Let's take a look at this region in short read data and HiFi data. With short reads in the top panel, 84% of short reads map with a MapQ of 0 and are not shown by IGV, suggesting that the aligner is confused between SMN1 and SMN2. In HiFi data, with the standard alignment, MapQ is higher than with short reads, but a big portion of reads are misaligned between SMN1 and SMN2. As a result, this region could not be phased into 2 alleles. In the bottom panel, Paraphase realigns all relevant reads to SMN1 and phases them into complete haplotypes that span this 41 kb long region. It assigns the haplotypes to SMN1 or SMN2 based on the differentiating position. Then, it determines copy numbers of SMN1 and SMN2 and makes phased variant calls. HiFi reads are grouped by the haplotypes they come from. We can see that in this sample, there are 2 copies of SMN1, a truncated gene copy with a deletion of the last 2 exons, and finally, an intact copy of SMN2. Throughout the genome, Paraphase analyzes 160 groups of highly similar paralogous genes like SMN1 and SMN2, including 316 genes in total. Among these, Some examples of medically important genes and their associated diseases are listed in this table. Paraphase provides a solution for these genes, which were previously considered challenging to genotype due to the high sequence similarity. Paraphase is available on GitHub. Its input files are, first, a FASTA file for the reference genome, and second, a BAM file where the HiFi WGS reads are aligned to the reference genome. Paraphase produces three types of output files. First, a BAM file where reads are realigned and grouped by haplotypes for the regions analyzed by Paraphase. You can load this BAM file into IGV and visualize the haplotypes by selecting Group Alignments by HP tag. Secondly, Paraphase produces a JSON file that summarizes phasing and genotyping results for each region. Lastly, Paraphase produces a VCF file to report small variants called for each region of each sample. In the VCF, each column represents a haplotype or a gene copy, where you can find the genotype, depth, and number of ref and odd supporting reads per variant. You can also find in the info field the coordinates of the phase regions on each haplotype. Next, I'll go over a few applications where Paraphase is used to study different problems. In the first example, Paraphase is used to resolve the complete sequences of SMN1 and SMN2 in hundreds of individuals from 5 ancestral populations. The authors classified SMN1 and SMN2 haplotypes into different groups based on the sequence, and identified new sequence markers for a special hard-to-detect type of carrier of spinal muscular atrophy, the so-called silent carriers that carry 0 copy of SMN1 on one allele and 2 copies of SMN1 on the other allele. The second study uses Paraphase to resolve CYP21A2 in patients of congenital adrenal hyperplasia. This gene resides in a 30 kb long repeat with common copy number changes, and it has a pseudogene that is similar in sequence. Each allele usually contains 2 copies of the repeat, one encoding CYP21A2 and the other encoding its pseudogene CYP21A1P. In this patient sample, reads from all copies of the repeat are realigned to one location and grouped into haplotypes. Paraphase phases the repeat copies into 2 alleles, indicated by the 2 colors, green and purple. The two copies in green are on one allele, with 1 copy of the pseudogene and 1 copy of CYP21A2, which carries a pathogenic small variant labeled by the star sign. On the other allele in purple, there is only 1 copy of the repeat, indicating a deletion, leaving just 1 copy of the pseudogene on this allele. Paraphase successfully solved this case and many other patient samples, reporting biallelic variants of CYP21A2. In this last example, the authors used Paraphase to identify de novo variants in trios by identifying haplotypes in the proband that are inconsistent with the parents. Here is an example of a de novo gene conversion. This is a group of 2 genes, the 4C gene and its paralog 4H, and reads are realigned to 4C. Haplotypes are shown in the proband father and mother. Each individual has 4 copies in total. inherited haplotypes are labeled in the same color in the proband and the parent. The second haplotype in the proband, labeled in red, is a 4C copy and was inherited from the second haplotype of the father, also labeled in red. This haplotype of the proband has an extra variant, denoted by the arrow at the top. The gained variant is from another haplotype of the father, the one labeled in magenta, which is a copy of the 4H gene. So this is a case of non-allelic gene conversion from a gene to its paralog. That is, a piece of sequence is copied from a gene to replace the corresponding sequence in its paralog. This is another example of how one can use Paraphase to compare the sequences of paralogs and study the biology of segmental duplications. I hope this presentation helps you understand how one can use Paraphase to genotype highly similar paralogous genes. If you have any questions or need assistance, please feel free to reach out to me at the email address listed on this slide. Thank you.